Good afternoon, sir. Hello, sir. Please unmute your mic.
Good evening. Could you hear me?
Yes, sir. Yes, sir. Yes, sir. Yes, sir.
Yeah, good evening, all of you. Because of some
Thank you,
technical
sir.
issue, joining Zoom is not actually comfortable with me. Okay.
Okay, sir. No problem. Achha, the name of the speaker in the last session is the Professor Raja Kumar Anantha Krishnan, IIT Kharagpur. The title of his talk is the designing of simple molecules and materials for sensing application. Emphasis on sensing in living cells and gases in environment. So Dr. Raja Kumar was born in Tamil Nadu. He obtained his BSc degree from MS University, Tamil Nadu, MSc degree from MK University, Madurai, Tamil Nadu. Further, he received his PhD degree in chemistry also from MK University, Madurai in the field of surface chemistry applications for effective of heavy metal ions from aqueous system. He had guided a short R&D experience from the private industry, and then he continued post-PhD scientific assignment in analytical chemistry and method development for alternative chemical analysis in ice water in National Center for Atlantic and Ocean Research, Goa. He had served as scientist C until 2007 in NCAOR, Goa. He had participated in Indian Antarctic expedition in 2005 to 6 and contributed for the scientific data for Indian ice core research, which was accepted by IPAAC organization. He had joined IIT Kharagpur since 2007 and now he is serving as a full professor in the Department of Chemistry. His research interest includes the designing of inorganic materials, metal oxide, semiconductors, modified materials, hybrids, and nanoproteins polymers for photocatalysis and functional surface materials, gas sensing, or sensing of toxic chemicals in water. He had guided 7 PhD students and 20 masters, including MTech. He published more than 60 articles in peer-reviewed journal and few book chapters. He served as associate editor of the journal Environment and Chemistry Letter, Springer, until 2021, and he was inducted as a member of NASI in 2019. He was a sponsored visiting fellow of Cardiff University through HEF CW GCRF Award 2019 and recently obtained EPSRC IIA Project Award 2020 to 2021. Thank you, sir, for accepting our acceptation invitation.
Yeah, thank you, Dr. Anand Kumar, for your kind introduction. So, good evening, all of you. I'm happy to join and discuss about my research and give some kind of idea about sensing of toxic pollutants like actually metal ions from the water or biosystems, or actually sensing of gas using a nanomaterials or a designed materials. This is my interest. So let me go to my topic and we will discuss more. Could you see my slide?
Not visible, sir.
Yeah, now you may be seeing that. Yeah, could you see my slide? Yeah.
Yes, yes, yes. Now it is okay.
Yes, that is best. So let me take the pointer and yeah. Good evening, all of you. Designing of simple molecules or materials for sensing applications. This is my topic today. I'm going to give a special emphasis on sensing of metal ions in actually live cells and also gases in the environmental gas mixtures. So I first of all I thank Atal program for giving this talk in the FTP at NIT Arunachal Pradesh. Okay, we know that day to day we are having a lot of commodities for our healthy and advanced life. We are using very rapid harvest of agriculture, We are making a lot of manufacturing products. We want to make a very colored materials. Nowadays, everything is under passion. So everything should be colored. Even whatever you are having, mobile cover, or you want to have a laptop, you want to have any material, it should be having a color. Even it is a polymer or it is a printed material or any other cloth material, we want to have a dyes. So a lot of dyes, pharmaceutical chemicals or other pesticides or agricultural compounds, agrochemicals, majority of the things we are using all are not actually safe. Majority of the compounds are actually leaching out to the environment. They are causing trouble to the environment, what is called pollution. So the pollution is nothing but introduction of any chemical or foreign compounds in the natural environment, which can create a harmful effect. So we need to know that what are the components are contaminating the environment. That is called monitoring. We need to know that how to remove or remediate these toxic pollutants from the nature. So this is one of my interest of studies, the removal of toxic pollutants.
Hello.
Yeah. So
Sir, is it possible to make full screen, sir?
yeah, full screen. I am keeping in full
Okay.
screen only.
Okay.
Yeah. Could you see the full screen or yeah, not it?
It is
So
not
you
full
just
screen.
configure. I'm putting full screen only. Okay.
Okay.
Fundamentals of toxic metal ions. Could you see the full screen? I'm in full screen mode only.
I
Okay.
think it is. Okay, sir. Okay. Carry on.
Designing a strategic fluorescent sensor, a chromogenic sensor. What is chrome? Chrome is a color, right? We know that a lot of things in the organic compounds or inorganic compounds, the color is one of the important actually aspect. In the inorganic compounds, we see that color is due to the metal means DD transitions, metal to ligand, charge transfer, or ligand to metal actually charge transfer. So, but in that organic compounds, where the color is coming from, that in the organic
Hello.
compounds, we have only
Hello.
homo lumo.
Sir, sorry, but hello.
Yes.
The slide also not changed, sir.
Is it? Okay.
Yeah, yes, because it is also not full screen and slide
Sorry
also
for
not changed.
that.
Okay.
I'll go back. This application actually I am not using the Zoom. Okay, let me see. Could you see my slide moving or not? Do you see me?
Sir,
Do you
sir,
see?
please change your slide. Please change.
Is it
No,
changing?
no, no. It's not
No.
changing, sir. No.
Let me try putting a
Okay, sir.
full
Okay,
screen.
sir. No, no problem.
Now could you see my slides are changing? Hello? Are you seeing the slides?
No, sir, you are sharing your screen.
Sir, don't share the screen. You just open the PPT in desktop and then share,
Yes.
sir.
That is what I'm doing, sir.
But
I am opening my PPT in the desktop and then I am sharing. That is what I
yes,
am
yes,
doing.
yes.
I have opened my PPT, the desktop. The file is open.
Hmm.
You are given a share access, right? So I have to share only
Yeah, yeah, sir.
You mean I want to share only the application, not the full screen?
Sir, you can share in the Windows also. There is a sharing option, na, sir?
Yes, I am sharing the window only. अभी मिल रहा है आपको?
हाँ, just wait, sir. Wait. It's showing. It is starting to share content.
Sharing a window, no.
No, sir. It is showing actually. Raja Kumar is starting to share content.
No, I already shared it.
But it is not
Connections.
showing as.
I'm sharing and then I'm changing the slides also now.
Sir, is it possible you just send to me and I will share here?
Okay. So let me see now. It is you are opening it or not? You are seeing or
No,
not?
it is not opening because it is going
Okay.
that showing that it is starting to share.
No, no, no. Yes. We are in the bad sync. I am opening the application, sharing. Once more, I try. If it is not working, let us go
Okay,
back.
sir, or you should disconnect and then join again, sir.
Yeah, something. I joined as a guest. Now
Yeah,
you are seeing my.
now it is fine. Now it is showing.
Now
But
it is
sir,
showing.
it is second slide. I think it is showing. So you
Yes.
just click in the left side. That third number. You just click in the third slide. Okay.
Yes.
And that means when you click in the left side in the number of the slide, that time it
Yes.
is changing. Okay, you can present in that way also. No problem.
Is it okay for you?
Yes, fine, sir.
Or I increase the size of the screen?
Uh, okay, just now this is also okay, sir.
Okay.
No, no
Okay.
problem, sir. Okay.
I regret for the inconvenience.
Oh,
Okay.
no, no problem.
Let me go to that. So I told a lot of chemicals and agrochemicals, dyes and pollutants reaching the environment, which causes trouble to the nature. So we want to do that monitoring and removal. So this is the aspect of our aim. So now we want to do design. You have sensor. In the sense, actually, when you want to go for a sensor, fundamental aspect is actually the fluorescent compounds normally we take as a sensor, fluorometric sensor. So the chemosensors can be actually different type, which can be the protonation or deprotonation function or redox function, or forming a complex with our target molecules or target metal ion, or breaking or making a covalent bond. These are the four aspects actually we take any chromogenic or fluorogenic probe. I told in the metals, we normally have a DD transition or metal to ligand charge transition kind of transitions. But in the organic compounds, normally the color is due to the HOMO-LUMO transition. So HOMO and LUMO molecular orbitals, the electrons are actually getting excited or de-excited through the actually the fluorescent process. We see that actually the color fluorescence. So what is the advantage of going fluorescence sensing? So fluorescence sensing is actually mainly selectivity or having high selectivity and sensitivity, and it is very cheap. One can easily use a fluorescence sensing in any kind of environment, whether it is a field or in the laboratory. So it can do a fast response, as well as actually we can do the local observation. So this is the technical advantage of using fluorescence sensing. The fluorescence sensing, there are two types are there we use. Normally is photo-induced electron transfer, PET kind of sensor. PET is a process, photo-induced electron transfer, or photo-induced charge transfer, either electron transfer or charge transfer. That is called PCT. So there are these two types actually major thing types we are observing in the photochemical prospects of fluorescence. Now we see that what is PET. The PET, there are two type again, actually two subcategories we can see. One is if you see that you are having a fluoroprobe, okay, fluoropore or a fluoroprobe you are having, but actually you are using another system, free receptor. If you see the free receptor which is interacting with your fluoropore, so then we will see the weak fluorescence. What is happening? Your main fluoropore is having HOMO and LUMO, okay, energy levels. When the free receptor, HOMO electrons of the free receptor is transferred to the HOMO of a, because what is happening, the energy gap between the HOMO. When the electron transfer will occur, whenever there are two energy levels are similar in energy, so they are actually comparable in energy, or they are having a similar kind of symmetry, then there is actually overlap will occur and bonding will occur. But in the photochemistry, what will happen if the two energy levels are comparable in energy, the electron from one level to another level can jump. So this is what happening with the free receptor with the HOMO. The electrons can come to the HOMO of fluorophore. So in another case, what will happen if you have a HOMO is lower than, actually the receptor HOMO is lower than the fluorophore HOMO. In that case, what is happening? There are two effects can occur. One is the emission from the LUMO to HOMO. The electrons are coming down. At the same time, the electrons from the HOMO of receptor can go to the fluorophore in the presence of the light. So which is only possible when we have a bound receptor. So what is the meaning that if you are having fluorophore and mixing with another reagent A and B, so that is a condition left hand side. The right hand side one is actually you are having a fluorophore and your receptor bound together through a chemical bonding. Okay, fine. So let us see if what is called actually PCT, photoinduced charge transfer. If you want to see the any kind of charge transfer, normally we see conjugate molecules. You know that actually benzene, naphthalene, okay, anthracene, kind of continuous conjugated molecules. What will happen? A lot of conjugations are there, double bonds are extended. The lambda is going to change. What is happening? That charge is getting continuously transferred. The same phenomena we take it here. We have a molecule. Yes, middle molecule is conjugate molecule. We are having left side and right side. We are having a donor and acceptor. The donor molecule is supposed to give a push effect, the pushing of the charges. That means actually what the electron releasing kind of functionality is supposed to be the donor side. If we are having another side, right, left hand side, right hand side, donor, left hand side, we are having, okay, of the molecule I am talking, left hand side, we are having a pulling side, that is the acceptor side. Acceptor side, the molecule is supposed to take the charge. So this kind of system when we develop in conjugation, which can do that ICT. Okay, so this is intramolecular charge transfer, because this system is after making that become a donor, yeah, conjugating mediator, and then acceptor. This entire system is going to have a single molecule kind of a macro system. So now this is going to have a intramolecular charge transfer mechanism. So we have a lot of examples in literature. You see that molecules are shown here, the boron containing heterocyclates. So we are having mercury. The mercury is going and binding with the nitrogen containing sites, nitrogen and oxygen. You see tetracoordination is happening here. So this mercury is getting bound. The same way, the examples are known in the literature, because the mercury, you know that normally we can form a linear, okay, coordination or tetracoordination. So again, actually one more fluorophore I showed in the second example, which can also coordinate with the ligand molecule, can form a coordination. So what is happening when metal ion is getting connected with this kind of ICD kind of a fluorophore? So what is happening? So there are two effects possible. One is, if your metal ion, you are designing with the donor molecule. I told the right hand side, the donor, if you are designing to capture the metal ion by a coordination bonding. If the donor is getting captured with the metal ion, okay, there is a MD coordination, MD bonding. Then in that case, what will happen? The energy gap between the HOMO and LUMO of the molecule after binding with the metal ion with the donor, the energy gap is going to increase slightly. That means the delta E, the energy gap between the HOMO LUMO, is going to increase high. So E is proportional to the HC by lambda, right? When E is increasing, the lambda going to be decreasing. That is what it is going to give a blue shift. So we are going to see that blue shift in that case when the metal is attached to the donor. If in case you are designing a molecule, the left hand side acceptor is the molecule going to bind with your metal. If your design is having acceptor based coordination, so in that case what will happen? So that acceptor and metal is getting bound. So in that case you are going to see that decrease in the delta E value of the complex system. When we are making interaction with the metal, the complex formation, after the complex formation, the delta E between the HOMO and LUMO is going to be decreased. That means the lambda is going to be increased. Okay, E is equal to HC by lambda. So the lambda is increasing, that is called redshift. These all the examples are given for metal related actually fluorescence sensors from the literature. So let us go in detail. So more examples if you want. People are also using materials. So TiO2, I want to take TiO2 bound with a probe, or you are having silica containing mesoporous silica, or actually silica containing substrate. You are attaching the fluoroprobe. So how to design these kind of molecules? There are certain tips I am giving in the right hand side. What are the requirements? The molecule or ligand you are taking, suppose here you see N double bond N, also kind of functionality. It's supposed to be a dye kind of molecule, or a molecule supposed to give a fluorescence after making a metal complex. So we should have a very careful screening of the molecules. All the molecules may not be a fluorescent probe, because the molecule itself should have a color, or it should have a fluorescence, or it should create a fluorescence after bonding with a particular metal ion. This is one of the criteria we have to take. So we can take a suitable nanoparticles, because what happens a lot of times, the functional groups present in the ligands can interact with the nanoparticles or the silica kind of substrates, can form a hybrid material. So this is the material aspects. Previous one I talked about actually molecular approach, ICT. Now I'm talking about heterogeneous materials type. So here you see people have used silica or A, B, C one I'm talking. The silica based or gold surface. People can create actually a substrate containing thiol group and attaching the gold, and another end, the gold will be attached to the fluoroprobe molecule. The molecule is already having a light absorption behavior. So these are the categories. Either we should have a anchoring group to attach on the nanoparticle, or a functional group which can easily attach on a silica kind of substrate, or we need to have a functionality to deal with actually gold or any other heavy atoms. So once you have this kind of molecules, what will happen? This molecule, you see the D, the example, we are having a molecule, the mercury is added. What will happen? The mercury will go to the nitrogen containing actually, nitrogen donor sites. The mercury is getting attached. Here tetracoordination is happening. So that fluorescent property is getting changed. So that will help us to do that detection of mercury. Majority of the fluoroprobes turn on sensors we talk it. What about turn on sensor? Whenever we have a particular target metal ion, we are having a probe molecule. When the probe molecule is actually getting bound with the target metal ion, that fluorescence is getting enhanced. So we call it turn on. That is enhancement in the fluorescence, or fluorescence is on. Turn off fluorescence is if you are having already fluorescence molecule, but when you are having a toxic metal in the water media or in the particular cell or blood sample, so when we are adding this particular compound on the metal ion containing media, so immediately the fluorescence is actually getting quenched. That is called a turn off. So what is a high advantage of these kind of systems developed in the last 10 years also? You can see that this is coming in Analyzed or Journal of Material Science or latest materials chemistry journals, Chemistry European Journal, or you can see that a lot of papers are coming in JMC. So this is mainly due to the high sensitivity. So stability of the molecule will be easier because you are having nanoparticle. Okay, you can take out and we can reuse. Can detect up to the PPT range. So I told the reusability. So these are the factors forcing us to design molecules which can bound with actually metal systems. So one of the examples I'm going to take with a small molecule. So designing of a chip based actually chemosensor for selective detection of mercury ions. So we know the chip base is actually popularly known, or actually people are knowing actually very much in the literature. So what is the ambition? We know that mercury poisoning from the incident in Japan, Minamata disease. In Japan, what happens? The mercury present in the ship sea is getting accumulated, bioaccumulated on the tissues of fish. So people take the fish from the Minamata Bay, got a mercury poisoning in their blood. So it created a lot of chronic diseases. So then later it was characterized that this mercury is coming from the battery industry, chloralkali industry. They discharge a lot of mercury in the sea near the Minamata Bay. So the fish normally a bioreceptor. What is happening? It is accumulating the mercury because any of the metabolite or metabolism taking place in the body and uptaking of toxic metal or any other metal ion, that will go as a bioaccumulation. So this bioaccumulation is happening like a natural process, because what happened, mercury is one of the simplest system which cannot which can go easily inside the cell wall. Okay, so it is getting absorbed on the cell wall of the fish, and then it is getting accumulated with the bio compounds present in the fish. So it is called bioaccumulation. Bioaccumulation means like a naturally forming a complexation. So this naturally formed complexation, what will happen if mercury is become mercury zero? That is actually pollution, nothing but respiratory pollution. If the mercury is 2 plus, is there mercury 2 plus? Normally, how it will give a problem? Mercury can be mercurous, mercury Hg2 2 plus, mercury mercurous. Mercurous can be a very simple system, or mercurous, mercuric and mercurous. Mercuric is actually Hg2 plus, which is going to be very toxic. So mercury, when reacting with the biosystem, form methyl mercury. Methyl mercury is one of the very toxic species people identified. That is what occurring during the biometabolic process inside the fish. Methyl mercury is created when people are consuming the mercury, that is creating a toxic effect to the people. So to sense this kind of a simple pollutants in the water, there are many methods are available. So we can say that a lot of chemical sensors are developed. Those are actually BODYP kind of chemosensors, calixarine based chemosensors, polymer based very tedious process of synthesis of polymers to do the sensor, or protein based sensing systems, DNA functionalized sensing systems, or nanoparticle based systems. Among them, small molecular sensor was very attractive to me. I was not aware that actually taking a very simple system can do the sensing of a toxic metal and very easy to demonstrate. However, later I identified that if the synthesis is not very tedious, it will not go to a very big journal. Okay, taking a small molecule and sensing and showing a lot of application is very, very interesting for people for practical applications. However, for scientists who want to publish paper, it is not very good. Because partially I am telling actually irony. If you want to do for sensing of mercury, you want to develop a very big calixarine molecule or very complex polymer molecule or prepare some DNA functionalized nanoparticle. If you do that, you will get a very big paper. But cost, the synthesis, synthetic cost, scaling up, application in the industry, these all the things are very challenging. Okay, so let us see that what people are doing for sensing of mercury. Biosensors for mercury sensing, chemosensors, okay, that we are doing actually fluorescence based sensors, conductometric sensors, or piezoelectric sensor, nano sensor. There are various sensors. What we have to see, the detection limit we have to see. The detection limit supposed to be 10 to the power minus 5 to 10 to the power minus 9. So which is actually one of the promising number for PPP level detection of mercury, because nobody want to do the 10 to the power minus 15 and all, because that much very low concentration is not going to be affecting the health. Because according to WHO, PPP level of 0.1 to 1 PPP level of mercury in water are going to create a hazardous effect to the mammals and other species. So we are looking for some sensor in that range, 10 to the power minus 6 or minus 7 or minus 8 or like that. Okay, let us see what is the literature evidence is there. So they are having actually, there are different examples. One of the Dalton paper 2016 I've seen from Yamato group. So they are using on chemosensor of mercury, okay, using water assisted, okay, heavy atom effect. So what is that ligand is there? Ligand and mercury, ligand is having actually fluorescence. When we had mercury, the fluorescence is getting quenched. So this, see that they have used 0 micromolar to 5 micromolar mercury like a titrating. So 3.48 10 to the power minus 8 molar. That is what I told. The lowest detection limit should be 10 to the power minus 6 to 10 to the power minus 9 will be highly preferable for practical applications. So this is the kind of a molecule they designed. You can see that a pyrene, okay, terminal group is there, and you are having other long chain attachments. So you need to have a lot of synthetic work to develop a molecule. But you remember, the molecule or probe supposed to have some light absorbing functionality. Fine. So let us go to the next example. If you see, develop a molecule like this, actually, you are having a very simple system, right? For synthesis chemistry, it's a very simple system. We are having a functionality like NH2. So we normally see that NO2 and NH2 functionalities are there. What will happen? This is my interest. So I took this paper from Dalton again. Kim et al. Has published. So here they have shown actually the detection limit is 0.57 micromolar. Okay, when they are adding the mercury, the intensity is increasing. That means this is turn on sensor. Okay, so let us go. I have told two different variety of sensor. So let us go to our work. So how to rationalize the design of a donor acceptor fluorescence chemosensor. What is donor acceptor? I had already told you. A middle one mediator molecule is there, conjugated with a donor in the left hand side and acceptor in the right hand side. If you develop that kind of conjugate molecule, can create a intramolecular charge transfer. So we want to exploit that ICT principle using a mercury to on fluorescence on or off sensor. So we want to synthesize a small probe. Yeah, I call it L1 in my work, which is having donor pi acceptor system. There is a conjugated system. We want to investigate the photochemical properties, and we want to know the functional groups. What are the functional groups really we want to decorate? What is the role of the functional group? Okay, we want to investigate the fluorescent properties on different heavy metal ions, and especially I am interested about mercury because that is actually one of the toxic metal ion. So I want to understand the sensitivity of the reaction conditions, including solvents and addition of different reagents and the pH. So let us see how to synthesize the sensor. We know normally amine and aldehyde we take and prepare a simple chip base. So I took this molecule. Okay, so I synthesized this molecule. Okay, one. When we synthesize this 80% yield, we could able to get. So we are having NO2 here functionalized. Two COOH groups are there in the terminal of the chip base, which is simple synthesis of amine, NO2 decorated amine, and taking aldehyde, we could able to prepare and functionalized COOH groups are there. So we got a kind of a nominal chip base. So now in presence of the DMF or water system at pH 7, we saw that absorption of 365 nanometer can give a fluorescence. Okay, this is a basic. So the synthesized molecule is actually verified using NMR and other characterization to check that actually the imine, what we are talking actually imine functionality, two imine functionalities we are having. We are having actually different NO2 functionality and imine functionality. So we want to characterize the molecule using the NMR, and we also wait for actually C13, and we want to know the proton carbon correlation studies. And finally HRMS, we could able to see that the molecular weight of the compound M+1 peak 418. So everything is favored. It's a very simple compound. We want to prove that the purity of the compound using different techniques. That is usual. Okay, so let us go for actually the fluorescence studies. When we started checking with the different metal ions, normally we take metal ions, not actually randomly going. We take a sequence of metal ions. So in that sequence, we took zinc, cadmium, mercury. These are the three neighbors we took: zinc 2 plus, mercury 2 plus, cadmium 2 plus. When we saw the fluorescent property of this molecule, we could able to see the fluorescent property of the molecules are different. Even in presence of various metal ions we took, okay, the only the pure ligand was giving actually absorption around 510 nanometer. You can see, sorry, the fluorescence about 510 nanometer, that is designated as L1. Okay, different metals, zinc, cadmium, mercury, you can see the mercury is a green color one, which has a less fluorescent intensity. This is studied in presence of various metal ions like a monovalent or divalent naturally occurring, because what happened when we study the fluorescence in the natural water, there are a lot of contaminants will be there. Are wastewater coming from the industry like Minamata case. They reported that a lot of manganese and battery industry. So a lot of manganese and chromium also came along with the mercury. So people could not detect using a simple sensor. So we want to develop a sensor where actually the chromium or iron or manganese can create a disturbance to the sensing. So we created a mixture, artificial mixture containing nickel also, because the electroplating or battery industry may have a nickel. So the mercury is successfully identified using a quenching mechanism. So when fluorescent spectra was recorded, okay, PL spectra, you see only dotted line is showing the only ligand. The ligand with mixed with all other pre-composition of elements other than mercury. You can see the blue color one, which is having slight difference in the fluorescent intensity. When we added actually mercury of different concentration, when we added 30 micromole of mercury, you can able to see that a small, the quenched spectra, you can see near 510 nanometer. So that is nothing but showing that the effect of the concentration, mercury concentration increasing, that in a mercury concentration, the fluorescence is getting quenched. So this is nothing but turn off kind of a sensor, quenching sensor. Okay, so when we studied this actually in the normal neutral pH, that is a B, the figure B showing that neutral pH, what is the sensing is happening. When we studied in the condition of a basic pH, we studied 7.5 is a 7.45 exactly happened. 7.5 we tried to prepare a buffer and study this, and then 8 and then 9. So we could able to see that at the basic pH, we could able to see that the fluorescent property is getting enhanced. So when we see that actually increasing the concentration, it is getting quenched. So basic pH is getting enhanced. I will show you the details. So here the pH influence I can show. The neutral medium, you can able to see the control I have shown in the separate insert figure. The fluorescent intensity of the compound L2. L1 is nitro containing. So when we synthesized L1, then we converted a L1 compound into L2. The nitro group is getting reduced to NH2. So amine functional. So that L2 is actually when we synthesized, we can able to see that the fluorescence intensity when we are increasing the concentration of the metal ion and the pH. So this is a comparison. First one is actually the change of the pH at the different pH values. You see that pH values are varied. Okay, so different pH values, the L2 fluorescence is getting quenched. When we increased the concentration of mercury from 0 micromole, there is no mercury, to 1 equivalent of mercury, what is happening? The mercury concentration increase also decreasing the fluorescence intensity. Okay, we got these two information. So when we are having basic medium, what is happening? You see that at a pH 7.45, okay, from starting from 7 to 7.45, I'm showing the figure B. You can see that fluorescence intensity is increasing when we are going towards the basic medium. So we were thinking that really we are getting some clue. When we are having neutral, the fluorescent intensity is less. When we are increasing 7 to 7.1, 7.2, 7.3, 7.4, like that, when we went near 7.5, that is exact value we identified, 7.45. So we could able to see that the fluorescent property is getting enhanced. You can see the physical image for the neutral as well as 7.5. So that brightness is showing that fluorescence intensity is increasing. So the D curve showing that in presence of the different metal ions, what is the fluorescence? We have put a bar chart. You can see that L2, the compound L2 is actually NH2 containing functionalized this chip base. Mercury is having very low intensity. Okay, so you can see that mercury L2 plus H, mercury 2 plus, having high intensity. You can see very high intensity comparable to all other metal ions, provided, because it is a quenching mechanism, I said. However, when we are adding a hydroxide or we are using some base, creating the pH around at 7.45, the fluorescence intensity is high. So this shows that the sensing of the mercury can be possible in presence of all other metal ions using this sensor. So what is happening? Time resolved fluorescence studies will show you that what is happening with the fluorescence with the time versus counts. Only the ligand we have actually the red color one. When we have a mercury of 0.25 equivalents, that is blue color one. That pink color one is mercury 2 plus and L1, the third one. So you can, I put an arrow. If you are unable to see for easy purpose, I put an arrow what is happening to the fluorescence counts. So after that, you see that I am treating, this is without treating anything as such in neutral medium. The right hand side figure B, I can show you, just I am having 1 plus 2. 2 is nothing but actually TMA. Okay, so I am using tetramethyl ammonium hydroxide. When I am increasing the tetramethyl ammonium hydroxide concentration 5, 10, 15, and 20, when I am increasing, the fluorescence intensity is actually increasing. That is what's shown from this time resolved fluorescence studies. So I'm putting the summary of this, what is the results in the bottom table. So decay parameters of probe, okay, is shown here. So two different eta 1 and eta 2. The average is eta nanoseconds. We could able to see only ligand 3.25, ligand plus mercury when we are having 3.22. When I am increasing the concentration of the mercury, it is getting quenched. So this is one of the very useful information, that mercury concentration is increasing, that is quenching the system. So how to make an application? First of all, we were worried about how to make application. We made actually simple a paper plate template kind of a system. We took a paper template without mercury, and only ligand is coated on the paper. You are seeing the physical image, that is 0 micromolar. So we could see that there is no fluorescence image. When we increase the concentration of a mercury in the ligand treated paper, like if it is similar to the, I can tell you, I am not telling the what is the composition. This paper is special paper, but it is similar to your Wartman paper only. We prepared this paper using a special cellulose treatment. However, we are trying for a patent. So in the meanwhile, I tell you, it is similar to your Wartman filter paper. So we made the paper treated with the ligand first, immobilized the ligand with the paper, and then we added mercury slowly. You can see that even we are increasing the mercury, you are not seeing much difference in the fluorescence. But if you are turning that pH of the medium as a 7.45, you can able to see very prominent fluorescence. So this is what we have done using the NMR titration. You could monitor that actually the peaks in the NMR, 0 equivalent to 1 equivalent. We have increased the concentration of mercury. You can able to see. So here the chemical shift value of A and B. So I indicated what is A and B is two hydroxyl groups I am showing in this figure. This is the L2 nitro. Okay, NH2, sorry, nitro is converted into amino. This is the L2 system, L2 ligand. When we use the L2 ligand, we could able to see that what is happening to A and B, the COH functionalities with respect to the different amount of mercury, what is happening in the chemical shift. So we could able to see that. So this actually, this mainly, this mercury is getting bound with the nitrogen containing donors, okay, the immunonitrogens. And again, it is getting two more sites are occupied with the water OH groups. Okay, so these two COH groups are free. They become actually, when we talk about mechanism, I will show you what is happening. So we identify that Jobs plot to get actually concentration of mercury. We can able to see that it is 0.025 micromolar at a 7.45 pH value. So you can see the bottom, zinc was added with our probe ligand. Okay, at 7.45, the color is actually, yeah, greenish color, a light greenish color for cadmium. For mercury, it is very distinct color of blue fluorescence. So fluorescence property of the different metal ions are different, and mercury is distinct, and we can see that selectively we can sense the mercury, the combination of our zinc, cadmium, mercury, and other metal ion mixture also. So let us see that how we can employ, what is the advantage of this system. So let me go to the advantage by showing this image, then I will come back to that. So theoretically, we did a calculation. What is happening exactly? When the molecule is normal, which goes intramolecular charge transfer without mercury, I'm talking. When the molecule is getting attached with mercury, you say that this nitrogen and the blue color dots, these two are two nitrogen atoms. When they are getting attached to the mercury, what is happening is, I have not shown because theoretical calculation, they have not included the water binding. It is a tetra coordinated mercury. Okay, when mercury is added, that the two phenyl groups are getting twisted. So the plane of the mercury group, phenyl groups are getting twisted. So what is happening, which is preventing the ICT process. Okay, if you want to bring back the ITC process, what we have to do, deprotonate the COH group in the bottom. So that is the beauty of the design. Simple fluorescent probe, okay, ship-based probe. We are having mercury addition, giving a quenching, but it can be, the fluorescent property can be retrieved using a deprotonation. That means adding a base. So this is one of the simplest approach. So when we calculated using the DFT studies, the energy calculation, which proves that actually our proposal, this mechanism is acceptable. Okay, the ICT is prevented when addition of the mercury 2 plus, and again deprotonation helps the stabilization of the molecule. That is what what DFT does. DFT does a calculation of energy. Okay, so lowest energy, the maximum stability of the structure. So that is what we calculated. So now we got actually, so when deprotonating, we are getting that fluorescence on. So we took a HeLa cell, live cell studies I told. So how to do? First we treated the cell, the right hand side image you see, the cell viability study. That means after treating the cell, we study for actually 24 hours and see that whether, here see that 6 hours and 48 hours comparison I put it. So we normally see 24 to 48 hours and see that cell counts are existing. That means that particular ligand should not be toxic to the cell. Otherwise the cells will be getting, okay, cleaved. So we should not have actually cell cleavage. We should have a stability of the cells. Then once cells are stable, then we will use that actually as a template for other applications. So here ABC, first row you see that live cells, HeLa cells. They are incubated in the absence of any ligand or anything. Only just we incubated the cells in a proper biochemical condition. We are not experts. We did a collaboration with the Department of Biotechnology with Professor Ghosh. And then second row, you see the D, F, G. So where, what we did, we incubated the cell with 30 micromolar of our ligand L1, the amino ligand actually, we just incubated there. Okay, then later C, H, third row, I say the third row, H, I, and J, you can able to see the fluorescence is actually prominent. What you are seeing, because we are adding mercury in this stage. mercury nitrate we took and added 10 micromolar in the cells. You can able to see, first of all, pure cells in the row, then we had a ligand, then actually adding mercury. But fluorescence, so that created a very big query. When we submitted to the Chemistry European Journal, that reviewer got misunderstanding. We wrote actually fluorescence quenching is occurring because actually the diagram what we shown, when we are having a mercury addition to the ligand, the A reaction, you see that ICD is getting disturbed. Once ICD is getting disturbed, fluorescence is supposed to be quenched. So the reviewer misunderstood that here fluorescence is prominent. Okay, so fluorescence is enhanced. So this is against our proposal without actually understanding the concept what we want to disclose in the work. So what we did actually, lot of fluorescence sensors we have seen in the literature. People normally have a sensor probe, as well as they want to have a target they attach. After that, for bringing the fluorescence, quenching of the fluorescence is normal. Any fluorescence containing compound reacting with the mercury and for a metal ion and forming a complex, quenching of the fluorescence is normal, what we showed till previous slide. But what we are showing in this slide, enhancement of the fluorescence, that is abnormal. Because what happened, to create such a fluorescence inside the cell, people have other reagents like a EDTA or adding a strong base or different other chemical compounds inside the cell, which can create a side effect. What are the side effect? It can be a cell toxicity, or it can give a side reaction, lot of hydrogen bonding, because you know that cell inside lot of amino acids, proteins, and enzymes, everything is available. So a lot of side reactions are possible. So in this study, only the ligand is kept. Okay. So what we did actually later, we did actually mercury immobilized cells we collected. Okay, HeLa cells treated with mercury. Okay, for about 6 days. After that, we took that particular things, cells, and then we treated with our ligand in a reverse way. Earlier, in this diagram, what I told the second row is treatment with our ligand, then adding the mercury. What we did in the final reversal, we did mercury immobilized. That means bioaccumulated mercury inside the cell, we took it. So those cells, it took it 6 days for us to collect the cell to get the mercury inside. Then we have added our probe from outside. After immediate addition of probe in the confocal microscope, we could be able to see that, yeah, promising fluorescent came. So the scientists, they operated the microscope. They told, even they did actually 3 or 4 probes for with the metal ion in the last 1 year also, they could not see such a brightness. So we could able to see again the beauty of this probe is, which takes that excitation wavelength 405 nanometer is visible light. Okay, not actually many times we go for a excitation wavelength in the UV light, which can also create a harmful effect to the cell. So this design, simple, small molecular chip base, is actually life friendly or bio-friendly compound, which can able to work with the 405 nanometer normal light, can create a promising prominent fluorescence. It's one of the beauty of this art. Here there is nothing is required to tune the fluorescence, because our design, I already told, there are two COOH functionality at the end. So when they are getting deprotonated, they are giving fluorescence. Inside the cell physiological pH itself helpful to deprotonate our probe molecule. That is a beauty. So automatically we got the enhanced fluorescence. So this is what occur inside the system. Okay, you see that the molecule, okay, having the fluorescence, the molecule with mercury is quenched fluorescence. When we are having adjusting the pH 7.45, we are getting actually the fluorescence is getting enhanced. So this is a charge transfer of the ligand is very important thing. The binding of mercury is one of the important thing. So what is the binding constant? We calculated, we have shown in the Jobs plot, which is one of the very good association constant we could able to arrive for this. And it works at around 7.45 to 7.55, so close to the very physiological pH, with the turn on up to 50 nanometer stroke shift. Whenever you are designing a sensor, you have to see that how much nanometer shift is occurring, enhancement. So it should be about actually 50 to 100. Sometime, okay, people put 70, 80 nanometer. So we got 50 nanometer stroke shift. So this work actually went to a good journal. Finally, it got actually approved to the chemistry select. So the donor acceptor system was very distinct for selective sensing of the mercury. I told the association constant value is 1.6 10 to the power 5 per mole, and the lowest detection limit 1.55 10 to the power 10 to the power 5, okay, per mole, and the detection LOD is 0.025 micromolar, which is which can be immobilized on the surface of paper like a cellulose containing material, or which can be used inside the live cells like a HeLa cells at the physiological pH. This is advantage of fluorescence sensing of mercury in such a system. Okay, I'm going to do that next topic, because switching the topic to different topic is difficult. However, I try my best. So one is a molecular sensor. I told, now material sensor I'm going to do. What is chemical resistor sense? Okay, chemi-resistivity is one of the phenomena we are going to use for doing the sensor. Application of chemi-resistive sensors are used in the environmental monitoring, okay, medical field applications, detection of chemical warfare, the safety, flammable, okay, gases, fire alarms. Okay, we are seeing that a lot of fire alarms in the industry. Okay, even modern buildings are having fire alarms. Okay, food and beverage industry, a lot of sensors are used for doing the adulteration. Okay, analytical approach. Automobile industry, already exhaust pollution detection. They are having alarms. Okay, the chemical resistivity of gas sensing is one of the developing field. What is happening? The surface induced phenomena. Here it is working. I will go the detail for you. Normally, you know what is called semiconductors of the materials which have band gap conduction and valence band. Okay, insulators have a very huge energy gap. Semiconductors have a nominal energy gap. Conductors don't have any energy gap, that immediately there is electron conductivity will occur. That means conductors are metal, you take it. Insulators are non-conducting. Semiconductors are partially conducting. How to conduct? We have to do some effort to push the electron from valence band to conduction band. Okay, so this is what happening normally. Semiconductor having energy gap around 4 electron volt. 2.5 to 4 electron volt gap is used widely for photochemical and other applications. So I work on that, but it is a additional work we did. What is chemi-resistivity? What is required? So chemi-resistivity is using a material is advantageous because high sensitivity and a rapid response, which can be easily developed in a gram scale and which can be employed in a industry. So Chemical Society Review 2019, you can see that what are the major things about the importance of chemi-resistive sensor. Okay, let us go to that. So we know that when we are using the material for a photocatalytic application, semiconductors, we have a conduction band and a valence band creating a hole and the electrons after shining a light on the semiconductor. This is what we know. But what we do that actually, this chemi-resistivity, we have a semiconductor metal oxide. If you take the surface, we are going to give a charge layer separation, where the space charge layer we call it, because what happened when this semiconductor is subjected to a particular process which is going to give a surface charge separation, like in the photochemical approach, the charges are separated and in the surface of the semiconductor. Here similar way, using electricity, we are going to create a charge layer separation. Once the charge layer separation is occurring, the surface will immediately respond to the incoming gas, so which can be oxidizing reaction or which can be reducing reaction, nothing but redox. Okay, you can see that G et al. Has published a very decent publication in nanoscale about how the chemical resistivity is working with the Schottky barrier semiconductors. Okay, so what are the crucial steps we want to learn? Surface and electronic property of the semiconductor is a main role, and geometrical factor. To tune the surface, we need atomic configuration or surface area or size or shape of the nanomaterial can play a role. This is what we learned from the literature review. So electronic structure means a lot of things are there: band gap, band edge, defects, doping. So many things are involved in the electronic structure. Geometrical structure means surface area, surface atom, or shape or size. So these crucial factors, if you remember, you can develop a new sensor. Okay, how people are doing, taking a p-type and n-type semiconductor. If you merge the second diagram, you can see what you will have. You will have a junction. The junction is called heterojunction. We have a junction means, what is junction normally? lot of trains are coming and getting connected in a particular station is called a junction, we say. Here what is happening, two different semiconductors, n-type and p-type, when we mix them, we are getting a particular in between property, that location is called heterojunction. Okay, apart from that, semiconductor can be hybridized with the carbon kind of materials, so that can form a hybrid. So what we really want, we learned from the Wang et al. Chemical Society review. We have published a paper previously in 2015 about the gas absorption capacity of semiconductors. We have studied another work in photochemistry and photobiological science, carbon and semiconductor, how it can create a charge separation. So we took this three combination of paper for reference to start the work. So surface activity and electronic property of a semiconductor is one of the aim. Second thing is we want to promote the charge separation so that we can able to do the sensing of the gases. So what people normally does, so methods for controlled synthesis of nanomaterial. What is controlled synthesis? Very simple, right? What is controlled synthesis? Taking a bulk material and going into the powdered material, then going for a nano. This is called actually top-down approach. Otherwise, you take atomic level, like vaporizing the atoms and the condensing the atom in a particular vacuum tube and converting into the clusters or molecules. Those things are actually bottom up approach. There are two approach to reach a particular nano dimension structures. So this you can see that recent development people are doing either top bulk material to do that or cold vapor or vapor deposition methods. So now we want to design a desirable size and shape so that we can do the particular property enrichment. So one of the important things I show you. So normally people do the different structures. You say for example, silver phosphate system, one of the systems actually people have used for various applications like semiconductor or photocatalytic or supercapacitor applications. So how it goes, I show you. So if you want to synthesize the material, I will show one example how that control synthesis goes. If you want to synthesize a material, take that actually silver nitrate and convert it into phosphate, right? You want to have a sodium hydrogen phosphate, you want to convert into silver phosphate. This is one of the approach. So what we did actually, we took a different templates like amino acids, glycine, alanine, proline, okay, hydroxyproline, and glutamic acid. So different compositions we took and we made a recipe of mixture. So individual compounds and mixture gave a different kind of results. We could able to synthesize the nano silver phosphate material. I show you here the genesis of the particle. I show you what is actually called top down approach or bottom up approach, how it is working. So when we started seedling of seeds, we are putting growing like a tree. So we put a seedling, we silver phosphate actually with a different porosity and morphology happened with a different temperature. So silver nitrate we took and sodium hydrogen phosphate, and we did actually a slow hydrolysis method, wet chemical method to develop. So every time interval we took a TEM analysis, SEM and TEM analysis. You could able to see the particles are small dots. You can see a big circle, big sphere is forming. Then again, very big sphere is forming. Like after that, you can see like a big laddus are forming. The small bundias inside, outside the surface. So it is what really happening actually. Aggregation of small nanoparticles and forming a big structures. Okay, what is the importance? It's a time is one of the factor and temperature is another factor. When we process with the temperature, different temperatures, what is happening? Some dissolution is taking place. That is called, here we call it as Oswald ripening. Oswald ripening means when the particles inside swallowing that smaller particle by bigger particles and creating a small porosity. You were able to see that in the last image, you can see small white color brightness is there. That is nothing but a porosity in the middle of the sphere. So such a spherical systems can be prepared by treatment of etching or temperature, Oswald ripening or hydrolysis. So based on this, actually, we could able to design different structures using different templates. This is example I'm telling. Different amino acids can give a different structures. So basically, what we did actually, we took a different amino acids like alginic acid or amino acids could give a different structure of the nanomaterial of silver phosphate. However, we took a mixture similar to the protein colloid. We write it BG. That is nothing but our artificial mixture containing all the amino acids of different ratio, create a protein colloid. That protein colloid mediated, that is like a templated silver phosphate synthesis, gave a microspheres. These microspheres showed enormous absorption of gases. So the studies of gas sensing is going on progressively with this material. So results are not actually concluded, so I'm not showing that. I'm showing for the next interesting things what we have a final results I'm showing in this topic. So we synthesized cobalt oxide CO3O4 octahedra from a coordination polymer route to get this nanomaterial CO3O4 octahedra. Then actually we studied for sensing of hydrogen gas in the atmosphere of toxic NOx. Okay, you know that normally sensing of hydrogen is very tricky. When a lot of gases are present, sensing of hydrogen is failing. That is why a lot of industrial fire hazards are taking place, because hydrogen is selectively sensing of hydrogen is very, very difficult. Okay, in the presence of other gases. So we want to design a sensor. So what we did actually, we want to prepare a cobalt oxide CO3O4, and a very specific cobalt oxide we want to prepare by a controlled method. So previously we showed controlled method can be done using some kind of template. Here what we did, we took a coordination method. Okay, we took a 2,5-DHS. Okay, so dihydroxysilane, and we saline compound and BDC. We took a BDC, benzene dicarboxylic acid. We took two different ligand mixed ligand systems, and using cobalt acetate, we prepared a complex. The complex is coordination polymer. But slow hydrolysis of the coordination polymer with the hydrothermal method, we could able to get a CO3 octahedra. Once we got a CO3 octahedra, because why I want a CO3 octahedra, when I prepare a normal CO3 O4, that is actually cobalt oxide nanoparticle, which does not have a particular planes exposed, 110 or 111 planes exposed. So we want to expose the particular plane, so we need a specific dimension of the nanomaterial. Once the particular faces are exposed, then we can able to decorate. So here PEOS, we took actually silica containing reagent. So we want to decorated the surface of the cobalt oxide. So SiO2 modified CO3O4. So we have a different ratios how to prepare. We have shown the different cobalt ratio how to prepare. This is very simple. We did a characterization of this cobalt oxide, we could able to see, and also FTIR show that SiO, silicon oxygen and cobalt oxygen stretching frequencies are visible from FTIR. BD surface analysis show that, okay, the pure powder cobalt oxide prepared by other methods, and our method using a coordination polymer method and modified with the silica, that we call it COSI 71. Our material is COSI 71. That you can see that BET absorption studies show that COSI system is having high surface area. So when we are designing a gas sensor, one of the property I told, surface property should be high surface area. So now high surface area, high chance for reaction. Okay, so we can able to do the sensing. So sensing is happening through the redox process. Now you can see that our synthesized material CO3O4, I told the octahedra. So now you see the octahedra designed with the Si silica. You can able to see that characterization is done with the TM, and 110 action, that particular direction the face is exposed, where we could able to do CO3O4 containing 110, we could able to decorate with the silica. Okay, silica on the surface, that is very, very important. So we went for sensing of the gas using the chemiresistivity method. So you can able to see the hydrogen gas sensing at a different temperature. This is only CO3O4 powder by normal method. By other people have reported CO3O4, the same CO3O4 we prepared and sensing the hydrogen. When we developed actually nano coordination polymer route, our route CO3O4, we could able to see that the sensing is having a lot of stability. Okay, it is not very rapidly getting actually absorption desorption kind of phenomena. Here it is having very good response at the 225 degree. You see that there is a huge gap, that nothing but the time seconds, how long it can show the response. This is what we recorded at the resistance versus time. This curve is showing the response. The COCA is showing very promising interval of response with respect to the time. The response percentage with various temperatures calculated. Temperature means 175, 200, because when it is a hydrogen is coming in the industry is not in the room temperature. A lot of reactors are high temperature. When hydrogen is leaking out to high temperature, it can catch fire. So we should have a sensor at high temperature so that we can able to prevent if there is any leakage of hydrogen. So we tried for 175 to 300 degree. We could able to see 175 to 250, 275. We can able to recognize the hydrogen gas of small concentration. So I will show you what is the response versus concentration. So you see here, SiO2 modified sensor is, you can see three different version. SiO2 modified sensor is having very small amount of detection is possible. Okay, so now hydrogen gas concentration I am showing from 20 ppm to 100 ppm. You can see like a linearity in the response. So very good linearity around 10 ppm. It could able to sense the hydrogen leakage in the system. So now with the time versus resistance, you can see hydrogen is showing very nice correlation with the temperature, sorry, with the time. So you can see that two promising peaks you are seeing. No, if the sensor is not very good, the peaks will not have very broadness. It will be like a sharp line. A good sensor supposed to have a very broad time gap. So we could be able to see at two peaks between 200 to 1000. That means the very good response and a slow sensing is taking place. Okay, so response you can see 250, 150, 200, 275 in presence of NOx. So NOx gas and hydrogen is mixed in this system. Still we can be able to see the sensing of the hydrogen is happening. So what we did actually, so what is happening on the surface, we created that a surface with 111 octahedra cobalt surface is exposed. The 111, what is advantage of this particular plane in the surface of the octahedra is helping oxygen to be reduced to O minus. Okay, when O minus is there with hydrogen gas, when hydrogen gas is released, it will form a RO H2O electron. This resistivity is measured in our system. We are having a small device, resistivity device, where it is kept under the vacuum on that plate. Then we are pouring the gas from the top, so it is showing the sensor resistance in the display. Based on that, we are going to. It's not very complex system. One can visit our laboratory and see what small system we are having. Okay, I'm not putting it here, but you can visit us or we can see our paper. So the silica modified system is having high response, high BET value. I told the hydrogen gas 100 ppm sensing. I'm showing 62.7. This is the response at 225 degree. When we see the normal commercially available or we prepared our CO3O4, it gives 3.2 percentage response. When we prepared a cobalt oxide with 1 plane exposed, it could show only 30 percentage response. The percentage response is 62 percentage. That means almost 30 times higher for the COSI. That means Si silica modified CO3O4 is one of the very useful system to show that enhanced sensitivity of sensing of the hydrogen. So we conclude that, so we could able to see the 100 ppm of hydrogen with the response of 3.2 percentage by normal cobalt oxide. Our system is showing 62 percentage. What is the reason science began? We want to create a redox surface 111. So when we have a redox surface 111 and coated with the silica in a uniform layer, then we could able to make the sensing of the hydrogen. Even it is very important, it is not a purely sensing only hydrogen. In the atmosphere of NOx, mixture of N2O, NO2, okay, NO. In that condition, sensing of the hydrogen is very toughest one. So we believe that this work will contribute for sensing of the industrial gases. So because of time, I am going very fast. One more example I will show you. This work is under publication, but I have a complete results. Nickel based another system we developed for sensing of the compound. So it is not actually very complex, but it is very simple. One-dimensional, two-dimensional nickel oxide we synthesized for the chemi-resistivity sensor applications. How we did, actually we prepared a one-dimensional, two-dimensional compound. Okay, we took a nickel again, actually nickel salt. We have a PVP. Okay, we formed actually two products. The product you can see that one is nickel DMG. You know that nickel, one of the important thing in the laboratory when we teach for students, nickel 2 plus can be determined using dimethylglyaxine. When we add a dimethylglyaxine with the nickel and add a small amount of base, it will give a very bright uh, rosy red precipitate. Okay, that can be gravimetrically estimated for nickel. So this DMG approach we took, actually ND, we write ND is a DMG complex we prepared, and also the BDC we took dicarboxylic acid, benzene dicarboxylic acid. We prepared a complex with nickel. So these two products are having two different kind of shapes. You can see the FESCM image. One is a rod kind, one is like a flower or leaf-like kind of a phenomena. So we took a TGA curve for stability. When we saw the TGA, we could able to see that both the systems are having stability. ND is degrading very faster in the with respect to the temperature, but a NBD slightly stable, more stable. So we characterize the nickel oxide using a powder XRD and other techniques. This is actually our analysis of a material using a TEM image. ND material is showing that material having small particles. You can see nickel oxide in the particular small composition. You can able to see that actually HRTM image show that hexagon kind of shapes, okay, which is under 50 nanometer size. So it is expanded with the 110 plane. We can see that facets. Why the shapes are important? I told the shapes. You can see that the shapes are having sides are having one plane. This side is having in this hexagonal shape 100. The top layer, like you can see that here, many places you are seeing like a okay 5-membered ring kind of structure. You are seeing that is 110 projection. So here we have 111, 110. Both the projections are available, which will form a FFT pattern. Single crystal XRD, if you do that, you can see the crystal pattern. Here, crystallite D spacing we calculated 002. Okay, this material we took it for sensing of the gases. Before the nitrogen absorption study show that surface area of this prepared material using ND and NBD both are actually 24 and 39. Okay, normal nickel oxide if you prepare 16 meter square per gram surface area you will get, but here enhanced surface area we are getting. Apart from the shapes, surface area also one of the important factor, so we want to count. So we did a XPS study to confirm that nickel is containing nickel or nickel oxide. So we could able to see that nickel 2p and oxygen 2p are confirmed using XPS photoelectron spectroscopy. Then we went for sensing of the volatile organic compound like ethanol sensing. So sensing of solvent, right? Earlier we showed gas, now vapors of solvent. In the laboratory, normally we get a fire hazard because of vapors. So sensing of vapor is also one of the important things. The nickel oxide able to do that. Sensing of these vapors at different temperatures. Here 200 to 275 degree, we showed that sensing ethanol. And you know that actually every peak is what is showing is sensing as well as recovery. So even you go for continuous sensing with changing the temperature, the individual peaks are appearing at a different temperature. That means the sensor is at it itself getting okay on and off kind of phenomena. That is what we say recovery. The recovery time is very important. Any sensor is going like a straight line, which is not showing any kind of difference. Once the recovery point is showing, then we will see the meter reading at the different temperature. The sensing efficiency can be calculated. So the bar diagram show that the sensor efficiency, normal nickel oxide, okay, it is a time, it is a given time, recovery time, you see that recovery time of a sensor of NBD and ND and nickel oxide at different temperatures. Okay, see the recovery time of an NaO and the recovery time of ND and NBD. So what we are seeing overall response, the normal nickel ND system is having 46 percentage okay response. NBD system is showing, because one dimensional and two dimensional there is a difference, so two dimensional system showing 49% recovery. However, normal nickel oxide prepared by our method shows only 35%. So the difference is actually then not only one temperature, we have to do the different temperature. We could able to see that always our NBD system, that means by benzene dicarboxylic acid routed synthesis of nickel oxide system showing better efficiency, mainly we told, which is a 2D structure having the surface planes, both the planes 110 and 111 are exposed. So the reason for the selectivity or sensitivity of the sensor is due to the okay found to sensitivity for each sensor is found to decrease, okay, but recovery becomes much faster. So recovery is faster means I told the gap between the peaks, the peak is coming down and increasing, that is actually become faster. If the recovery is faster, the sensor can sense back to back release of the more amount of the vapor. Otherwise it will not sense that actually release. Once it is giving alarm, the sensor will not give any other further response. After that, it will be exhausted. But our system continuously leak is not arrested. It will give a continuous signal. So continuous alarm will be coming. Continuous display reading also will be coming at different temperatures. So we are seeing that faster recovery rate is important. So thermal promotion is possible in this case. So at the end, we can see that performance of our sensor is given in this NBD and ND comparison recovery response. You see the NBD system is having more response than the blue color curve, linear curve in the last image, than the green color one, that is actually ND. So response and percentage transitions of VOC 300 ppm at 225 degree. We did actually ethanol concentration 300 ppm in the chamber, small chamber we prepared, and within the chamber we evaporated the ethanol 300 ppm we created. So in that atmosphere, NBD at 225 degree showing that, okay, the black color one is for ethanol, then we did acetone, the red color one, methanol, we have a green color one. So the same way we did for ND, black color is for ethanol, red color is for acetone, methanol is for green color. So the bar chart shows that the performance of sensing of ethanol, acetone and methanol, you can see that ethanol sensing is actually possible by ND and NBD. Acetone sensing is not very possible by ND, but NBD can do methanol also. So NBT is overall a better sensor for sensing of different volatile organic compounds at the temperature like 225 degree. It is not room temperature sensing is actually not a big deal, but sensing at a high temperature will be useful for industrial application. So when you have a queries why you are not doing the room temperature sensing is not actually our aim, because the room temperature it will not have a catching fire. Normally fire hazards occurs at high temperature, especially in industry and reactors. The leakage occurs that nearby surface temperature of the reactor is at around 150 to 200 degree. So we are bothering about fire hazard at the high temperature. Okay, so the ethanol sensing mechanism, how it is working, if you have oxygen atmosphere and reacting with the electron gives a O2 minus. So if you take a methanol, what is happening? CH3, C2H5O minus plus OH minus. So C2H5O minus, if you have a gaseous state which will absorb with the oxygen and give a C2H5O, C2H5 twice O. What is happening is we are only using that oxygen is getting reduced to O2 minus. Once oxygen is getting reduced to O2 minus, which reacts with the methanol. Okay, ethanol and forms the C2H5O minus. The way we are getting that the collection of electrons are coming out, the electrons are showing the resistivity in the display unit. The same way you can see the any kind of you take acetaldehyde also. So you take O2 minus, it will form a CO2 H2O and H10 plus. So this is the basic principle of sensor actuator showed 2017. The similar kind of mechanism we try to propose for sensing of the methanol. So in our home tech message, NBD system is having high surface area and could able to do the better sensing capacity. The recovery of the system NBD is much faster because of large pore diameter. So with this, I conclude that actually we could able to prepare a one-dimensional and two-dimensional nickel-containing architecture, and they are able to do the sensing of the toxic volatile, not toxic volatile solvents because of the open facets of 110. So we could able to see the good surface area values of prepared nickel oxides are helpful, and we could able to see the response of the system is actually much higher than the normal nickel oxide. So synthesis of a shape or a facet controlled synthesis of a nanomaterial is advantageous. This is one of the important message for beginners. If you want to go for gas sensing applications, one has to do the shape or no facet controlled synthesis is very very important. So here I conclude that the first topic I told fluorescent on off on sensor using a chip base for mercury sensing, which works under ICT, intramolecular charge transfer. The second work I showed is actually controlled synthesis of cobalt oxide, where 111 planes are exposed, which can be useful for sensing of hydrogen. Even you see in presence of the NOx, the ratio I have given, NOx 10 ppm, 0.5 percentage only interference, but 62 percentage hydrogen. That is actually quite 60 times higher sensitivity for hydrogen, the second system. The third system is one-dimensional and two-dimensional nickel systems. Nickel oxide systems are developed by controlled synthesis, which shows that sensing of the ethanol, acetone, and other solvent vapors successfully. So this work is under the revision in one of the ACS journal. So I will discuss more in future. So this is one of my favorite pictures I want to put you. Dr. Anant Kumar was telling about my Antarctic visit. This is middle person is myself. So I've been to the Indian Antarctic expedition in 25th Indian Antarctic expedition for collecting the ice samples for analytical chemistry and to study the climatic changes and the chemicals present in the South Pole Antarctica. So if there is opportunity, next time I will talk about this my analytical chemistry aspects of ice samples. So I now stop here with the thanks to my students who contributed, Mr. Alok Kumar and Dr. Subrada, who contributed for Alok Kumar contributed for mercury sensing, Subrada Mental, who is now working as a postdoc in Germany. He contributed for gas sensing application using nickel as well as cobalt oxide. But Rakibuddin, he helped me to understand the cobalt oxide system for gas sensing and gas absorption and gas sensing applications. I thank DST New Delhi, IIT Kharagpur, and GCRF Cardiff University for my collaborations. I also thank my collaborators for gas sensing application, Professor Majumdar at Material Science Center, IIT Kharagpur. I thank Dr. Anand Kumar Atta and his actually organizing team, NIT Arunachal Pradesh, for giving me opportunity to talk. Thank you, all the participants. If you have any query, I'm happy to interact with you.
Thank you, sir, for your nice lecture. I think in a short lecture you have discussed so many things. How we can synthesize the simple molecule and we can use for sensing purposes. Not only organic, small organic molecules, but also we can synthesize some materials, metal-based materials, and their use. So, participant, if you have any question, please ask.
Thank you.
Sir, actually, I
Yes.
have one query. So,
Yeah,
if you
welcome. You're
open
welcome.
the slide number 16.
Yes. Yes. Yeah. Please ask me.
Yeah, this one actually, sir, what solvent you use to detect the mercury? Because you told that your sensor as pH selective.
Yes.
So when it is 7.45 or 7.5, it shows the fluorescent enhancement.
Yes.
And in neutral medium, I think it is fluorescence quenching.
Yes.
So which medium you used?
Okay. I will show you. I have already shown you, but actually, I will show you again. Actually, what DMF water mixture, 2:3. Okay.
Okay. So
You can
and
see
what is the
this.
what is the function of the acid group to increase the simple
Yes,
solubility?
actually, normally this acid group is not required originally speaking, because in our mechanism without the acid group also the because we need to what we need imine nitrogen is required to bind with mercury. Okay, so the benzene rings are required for a conjugation purpose. Okay, what we
Yes.
did the acid group. You are very interesting question you ask. We carefully designed this acid group. Without that, we prepared several sensors. We could not publish. We could see that this help for anchoring inside the biological cell. If you don't have any polar functional group, we cannot do. So we
Okay.
have done
Okay.
actually three work. One is biological anchoring. Second one to anchor with the titanium dioxide, metal oxide. That I am not showing here in this work, but we did a work on metal oxide. How to anchor the metal oxide? We need one functional group like a OH or COOH. So we use this functional group for multiple purpose. So if there is a polar group, which can easily go inside the our system, or even actually we modified with the TiO2. TiO2 easily binds with the COO functionality. Okay, COO minus and TiO2 binds, and then we can able to send inside the because TiO2 is harmless. We can easily send inside the cell systems. So this design is actually I did. So if you are not asking, I will not have opportunity to tell this. Okay, thank you. Yeah.
Sir, another thing, sir, the use of DMF
Mm.
or maybe in DMSO in the living cell, but because you have taken the cell imaging, you have done the cell imaging study.
No, but
So,
actually, yeah, you are right. But actually, for laboratory experiment, we use a DMF water.
okay,
But
okay,
originally, in this paper, chemistry select paper, you can see we used only water because this actually this is water soluble. Okay, polar. Okay, we did studies with only water, not DMF. We have not taken inside the cell. Inside the cell, what we did, we added actually 30 micromolar of this ligand system inside the cell and incubated. Sorry, we did actually for culture for 48 hours. After that, we added the mercury. So we have not
Yes.
used any organic solvent because it will damage the cell. You are right. But
And
DMF is DMF 2:3 is not damaging. We did a control study. However, we are using neutral condition only. We are not
okay,
using
so
any other solvent. Yeah.
and there in the cell, inside the cell also you have maintained the pH that
Yes.
7.45. I think you have written that.
Yeah, but actually it is not maintained by us. It is nature. Cell physiological pH is 7.43. So it helped. We prepared 7.5 in the laboratory. Okay, 7.5 to 8 we did in the laboratory. But when we took inside the cell, it is default. The cell is having a physiological pH, right? So our collaborator identified and told 7.43 is the pH of the cell media. So we are not using any other buffer. When we prepared in the laboratory, we used 7.0 to do all the study. pH effect we want to study, we went for exactly 7.5 because in the lab we can able to prepare 7.5, 7.7 or 8 and all. But inside the cell we cannot do anything. So inside the cell is it was told 7.43 to 7.45. So we took average value. Okay.
Sir, you that means you have done the experiment, laboratory experiment in 7.45.
No, we did a laboratory experiment in 7.0 neutral.
Okay.
Then
Okay.
for fluorescent turn on, we added a base externally. You can see that in this diagram, you can see that, not here. Yeah, next one. You see that TMA, we have added TMA.
TM.
Okay,
Okay.
to make that actually this deprotonate the proton in the COH group. You got it. So this is
Yes. Yes.
another
Yes.
when we deprotonate, we are getting actually fluorescence. What happened? This ICT mechanism is hinted after
Disturbed. Yeah.
coordination with mercury. So when we deprotonate, again it is flipping. This configuration of the phenyl group, actually the benzene rings are flipping. So it is giving the turn on. Okay. So this is another application. Even not only COH, we can use the OH also. The same effect will occur. So we use actually COH, so we use to base here TMA. We use actually for activating. But inside the cell, the TMA is not required because cell itself having 7.4 because basic. The laboratory we did the first study is mercury binding 7.0. At 7.0, it is getting quenched. But at 7.45, okay, 7.45 to 7.7 we did a study, but 7.45, this is one of the example I am showing here. So usme, this actually giving turn on. So this both effect I have shown in the another diagram. You can see here, not here. Yes, here you can see this one actually. There are A, B, bottom two figures are there, no? Wavelength and absorbance. So pH 7.45 we created in the laboratory. We added HTMA to bring the 7.45 to study, and right hand side neutral medium only we could able to see. Both are giving a similar thing. Only in the presence of the base, the A figure A in the left hand side, giving the enhancement in the fluorescence. That means the fluorescence intensity is increasing at the basic pH. Because our one of the reviewer asked, you have done all the experiment at 7.5, but the cell is having 7.43. So how can you compare? So we did a study for 7.43, 7.4, 7.45 like that in the laboratory to compare with the physiological pH. Okay.
Thank you, sir.
Yeah, if it
That's
is not working, if it is not working in the basic medium, our system would have failed. So cell is basic in nature, so it is advantage without
So
adding. If
yeah.
other people are working in a HeLa cell for fluorescence, they are adding some reagent like EDTA, or they want to add a base inside the cell, again increase the pH like 8 or 9. So that will not be suitable for a living cell detection. So we are not adding any other reagent other than the ligand. The ligand is, is a ligand is a chip base. So chip base is again bioavailability, right? Chip base is safe. So if there is a mercury contamination is there, the chip base we are adding and just sensing the cell. We are not introducing mercury. So our intention is, if the cell is already bioaccumulated mercury, just adding our taking the cell, adding our ligand can do without disturbing the cell. So that is what this sensor actually was very much impressed by other people. Unfortunately, it was not going to the bigger journal because we are not expert in the field before this publication.
But work is excellent, nice actually.
Thank you. Thank you.
Okay, sir. Participant, if you have any question, please ask. Any question, please?
I'm happy to answer any question. You ask any question, no problem.
Hello, Dugnam is here.
Please ask question so that it will be help. It is a help for me. If you are asking question, I will be happy. Dr. Anand Kumar asked a
It
very
is
nice
also help,
question.
sir. It is help for me also.
No, mutual. It is mutual. Because the COH functional group, no, there when I am synthesizing this one, my student asked, sir, I don't want COH. It will be complex. I told you prepare, then I will tell you the reason. Then it was appreciated by the editor himself. Editor wrote, this work is very good in chemistry European journal. Finally, one of the reviewer told, no, chemistry select you can transfer. Otherwise, it will go to chemistry European journal. Yeah. But
Sir, except this, sir,
but
except this
I
presentation.
was recalling that your work lot of nitrogen containing heterocycles you prepared, right, Anand?
Actually, sir, I have started based on the carbohydrates because
Okay.
carbohydrate based sensor I have developed and published some more. So actually, we use the carbohydrates to basically increase the water solubility, and we can easily functionalize. Sir, here actually basically we don't have the so much research facility. So in 1 or 2 days we have synthesized the molecules and then characterized. And then we maybe measure the fluorescence or UV, and some cases also we have taken the cell imaging study. So
That is what I shared my failure. What is my failure is if you take very simple molecule, the world
yes,
will
sir,
not
yes, sir.
accept you. You want a very complex synthesis and a very big molecule, then you will get a very big paper.
Yeah,
But
yeah.
for useful application, one should think a very small molecule, very simple carbohydrate or very simple polymer.
Yeah, so synthesis process should be simple.
Then actually it will be very useful for industry. So we are actually thinking about a very easy layman chemistry. Anybody can follow that chemistry is impressing to me. So we did like this.
Okay, sir. Thank you. I think participant had no questions. So thank
Okay.
you, sir. Sir, thank
Thank
you
you.
for
Thank you
accepting our
for
invitation,
inviting
sir.
me.
Thank you, sir. Thank you, sir.
Thank you. Thank you. Best wishes to all the members. Okay. Thank you very much. Bye.
Thank you, sir. Thank you.